It is 7:30 and welcome to the March Valleywise Grand Rounds. As with other months with Valleywise, keep in mind Megan will be putting the CME code in the chat midway through and then again later on during the presentation. This month, we're very glad to welcome Dr. James this morning to discuss molecular testing and its role in our pediatric practice. Dr. James is a trained neonatologist from Stanford who started his practice in neonatology, then over 15 years later, went back and obtained his MPH in training in medical genetics from the Harvard Medical School Combined Program in Medical Genetics. Dr. James worked in Massachusetts until coming out here to the Valley, and he's been very involved in both research and in resident education here as well as in Boston. With that, I will hand it over to Dr. James.
Well, thank you, Rachel, for the introduction. I was kind of struggling as to what topic to do because there's so many interesting topics that I think have relevance for pediatricians and subspecialists when it comes to genetics, whether it be newborn screening, molecular testing, which I'm going to do today, dysmorphology. And I thought that because of the new state program for basically, I don't know if subsidizing is the right word, but a payment option for whole exome or whole genome screening of sick newborns on access, that I would do molecular testing and talk about the main tests that we all order on our shared patients. So this is on molecular testing. I don't have any disclosures, and I'm going to concentrate on the types of tests that are highlighted in blue. The old school karyotype, the chromosome microarray, a updated version of array, CGH, that kind of came on the forefront in the mid 2000s. And then gene sequencing, both whole exome and whole genome sequencing. This is just an overview where, we have different parts of the body with different tissues. They have different functions, but all cells have essentially the same genetic information, and what differentiates one cell from the other is which genes are active and which ones are silenced. A great deal of research done on that. The main thing here is that if we have a child who has, let's say, a seizure disorder or a problem with the cardio system, that we don't have to actually go into the organ of interest, that we can do gene testing on a surrogate sample like blood or the cheeks. This slide, a little cartoon showing the cell, the mitochondria, which have 37 genes for transfer RNAs, ribosomal RNAs, and a few of the electron transport chain complexes. But by and large, the 99.99% of all the genetic information is obviously in the nucleus on chromosomes. And I kind of liken this to a double necklace, a helix, double helix to parents, and talk about coding letters. And that when we have thousands of them in a row, we obviously have genes, and humans have roughly 20,000 protein encoding genes, as differentiated from non-coding genes. And the interesting thing is that if you add up all of the genetic information, like all of the base pairs for all protein encoding genes, this may be at most about 2% of the entire genome. So the vast majority of our genetic information is composed of non-coding DNA, either the introns, which are in this slide, the exon, intron, exon, or the promoter region, uh, or the 5' untranslated exon, and generally downstream from the termination codon are enhancers. There's things called silencers, and the neat thing is that there are huge stretches of genetic material from one gene, and let's say you walk down the chromosome, there would be another gene where I'm moving the little cursor. There'd be another gene down here, and somehow in the three-dimensional structure of our chromosomes, there'll be multiple chromosomes which will be in very close proximity to each other, where genes are simultaneously transcribed with common transcription factors. They'll be silencing through methylation of histones and other things. So that's just kind of a general overview. So the old school was to do high resolution, like 550 band karyotype. And we're seeing here a karyotype of a male a child. And so the karyotype is basically lining up the chromosomes by length, by bands. And the question is, is every band there like it should be? Is it oriented in the correct position? Uh, and there should be obviously 46 chromosomes, 23 from mom, 23 from dad. But the resolution of a karyotype is about 3 to 5 million base pairs, which was, the best that we had in the 70s, 80s, 90s when I first started doing neonatology. It was the only test that was available. And there were certain genetic conditions like velocardiofacial syndrome or DeGeorge syndrome, where there would be about a 2.5 to 3 megabase deletion of the lower arm of chromosome 22, 22q1, 1.2. You might be able to see that, but it was unable to resolve what we would call submicroscopic uh, copy number variants, either extra pieces, duplications, or deletions. So a common condition that we see is an infant or a child. I actually saw a child yesterday in genetics clinic, one of the Afghan refugees, where uh, they were diagnosed with Down syndrome. There was a suspicion of that back in the home country where the child was born, but did a karyotype. And this is obviously the standard trisomy 21, where there's 21 chromosomes. And this is a fluorescent in situ hybridization where there's a tag that is kind of like a Velcro that is complementary to a sequence on the chromosome, and it will highlight or shine under ultraviolet lights and give a signal, and there's different colors that are available. So you can see in this that this metaphase has two greens, and I have no idea which chromosomes these are, but basically the normal would be two for, let's say, the X chromosome, since this is a female individual. There are two blue, but there are three red signals, where the red was a probe for chromosome 21. And this is, one of those rapid metaphases that can be done on amniocytes, let's say, or on a sample in the nursery ICU. Uh, and the answer will come back in general faster than a chromosome microarray. So the neat thing about a karyotype is that in some respects it's giving quantity of genetic information, but it's not very accurate, like I said, because you can only resolve down to maybe 2.5 or 3 megabases. But the key is, where is that genetic information? So here we've got a a, a balanced translocation in a mother where you can see that there's actually only 45 chromosomes, 45, um, the XX is not shown in this, but you see there's only one 21 chromosome, but on the 14th chromosome, one of them, at the top, the P arm, there's a piece, an entire 21 chromosome here. So this mom has the correct quantity of genetic material, but it's organized in a different manner. So when she creates an egg through meiosis, it'll either be this chromosome or this one that will go in. It'll either be this one or, the one that's not there. And the same thing goes for all of these other chromosomes. So what happened in this scenario is that the father gave a normal complement of one chromosome for every autosome. So you can see two copies all here down to 13. The father donated this normal 14, donated a normal 21. The mother donated this normal 21. So here we have two 21s. But the problem is that it was this 14 chromosome, the hybrid chromosome with the Robertsonian translocation, that has the 21 chromosome on the top. That's the situation here. So there's one, two, three copies. So this is a unbalanced translocation in this uh, female because it's 46XX. There's 46 chromosomes rather than 47 in the traditional trisomy 21. The quantity is different, but the location is different. And this is important. And this is why it's most important to actually do a karyotype on a child that one suspects of Down syndrome, or let's say trisomy 18 or 13, because or any other kind of combination, quite frankly, because we want to know, is there a possibility of recurrence in subsequent children, and give that advice to the mother and father. As far as the child's health is concerned, where this extra 21 doesn't matter. The child's phenotype with Down syndrome is going to be the same as any other child who has the classic presentation. So here going back to exons versus introns. And typically, what we do is sequence the exons because those are the protein encoding regions. We also sequence the boundary, or what are called consensus sequence, like about 20 base pairs either side of the juncture of the boundary of the exons and the introns. But we don't check the introns, which are by and large a majority of the gene. So here we have like the gene, it's transcribed with RNA uh, polymerase, it's full length, and then you have splicing where the introns are looped out, and then it's translated in the ribosome, and you get a full length polypeptide or protein. And then once it exits the ribosome and maybe it goes through the Golgi apparatus, it's going to fold into the protein conformation that is active, and then it might be modified with glycosylation or other kinds of deals, and then sulfhydryl groups will happen. So in traditional sequencing, it doesn't matter if it's a single gene or a panel of genes like Noonan syndrome panel, what we call rasopathy disorders, or whole exome or whole genome, strike the whole genome. We're only doing the exons in these boundaries. And the main thing is we're looking for if this is normal with a G base in this location? Is it a T? And, you know, we're not clones of each other. So we're all going to have these so-called polymorphisms. The key thing, though, is the change significant? Is it a pathogenic mutation? Is it one that is not tolerated? Is it one that causes the encoded protein, which is the key thing, the protein having a specific job to do? Is that protein going to have a different structure or uh, biophysical uh, difference, like in charge or shape? Will it cause a conformational difference, different angles? Will there be amino acid groups that will not touch in a active site, or will it not interact correctly within the membrane, like in the case of neuron receptors like neuroligands or MNDA receptor, things like that. Or so that's that deal. I did DNA sequencing by a technique in 1977 called Maxim Gilbert, which was a real nasty, using a lot of toxic chemicals where you end labeled the DNA of interest, and then you chemically cleaved sequentially each nucleotide. So you'd have a ladder and you get something like this, uh, the ACGT, and you'd kind of read it up, and that was very old school. Then Sanger from Britain did the Sanger technique, which was using the dideoxy uh, bases, which basically caused chain termination, and that caused a ladder. So you would look at the electrophoresis pattern. But now there's high output, massively parallel sequencing, and this is some lab that just has, you know, tons and tons of these sequencers going 24/7. There's always new technology that's coming around the corner, and a sequencing run might only actually take a couple of hours or certainly overnight. And what they're doing is they're cleaving the DNA of interest into random pieces. And these pieces are basically overlapping each other. And then they're asking the question, how many of these A's, let's say in this line that I'm showing you, how many A's are lining up? And so statistically, if there's more than 20, let's say, there's going to be a higher level of confidence that that particular sequence is valid. Uh, you've probably heard of the American College of Medical Genetics gene panel. The current version 3.2 has about 80 genes, and these are all medically actionable disorders. And we offer this for our patients, for the parents, to say, well, does your child have a pathogenic BRCA A1 or 2 mutation, hereditary breast ovarian cancer in females, prostate cancer in males, higher incidence of pancreatic cancer? And I give the analogy of Angelina Jolie, who has on her maternal side a lot of women with breast and ovarian cancer. So she underwent gene testing and was found to have a BRCA1 mutation. She went public to educate the public that this is something that one could do proactively. She underwent double mastectomy and oophorectomy to eliminate the risk of her potentially developing breast or ovarian cancer. Now, this has no relevance for a 5-day-old infant, uh, but it is offered to families. Things like multiple endocrine neoplasia, aortopathies like Marfan syndrome, Lou-Dietz, other kinds of aortic aneurysms, the Titin gene, which is associated with cardiomyopathy, cholesterol, lethal long QT intervals, Pompe disease, there's Fabry disease, and OTC, which can have a late onset presentation. Here's, I'm going to breeze through this because this is kind of like the technique of going to chromosome microarrays now. So we can do it with a cheek swab, but this is just showing a blood sample, isolating DNA, and then the patient's DNA is, in quotes, tagged with a red fluorescent label, kind of like that FISH test. And then a so-called control DNA of someone who does not have a copy number variant is tagged with a green label. And there is a huge matrix uh, that has oligonucleotides that are about 1,000 base pairs long, which are in an array. They're in a matrix. So that's why it's called a microarray, because it's microscopic. The actual chip is, you obviously can see it with your eye, like a computer chip. And you react the sample that's mixed of control DNA and patient DNA, and you're looking for colors in this array, this matrix, and it's scanned. And so you can have a normal result where there's the same number of red and green, and actually it turns out to look yellow. You might have a gain in the patient, so extra reds, that looks red. And then you might have a deletion, and so then it has a different color. So chromosome microarrays are good for finding submicroscopic copy number variants, either a deletion like this, where there's a shorter chromosome that's missing a piece, only one copy, whereas everything else there's two copies. Here in a duplication on the terminus of this chromosome, there's three copies, the two here and one right there. The other neat thing about a chromosome microarray is that it's able to recognize if there's regions of the two chromosomes of the patient which are exactly the same. So in other words, if there's a mutation in one location, is it in the same location in the other allele, on the other chromosome? Yes or no? And there can be huge stretches. And so the closer that one is related, the closer the parents are related, there's going to be a higher level of what we call loss of heterozygosity or presence of homozygosity. And so if you have homozygous regions and you have within them a deleterious and negative pathogenic mutation in a specific gene, then there's a very much higher incidence that that patient will have an autosomal recessive disorder. So sometimes the chromosome microarray, although it may not tell you that the patient has disease X, it's going to give you a clue that that might be present. That's the normal one. This is just a scan showing like a baseline. And then here a deletion where it shows going down in quantity down to one. And then a duplication where there's a rise right here of a duplication of this particular segment. So kind of in conclusion, if you compare a microarray to a standard karyotype, what the chromosome microarray can do is it can, of course, detect large deletions and duplications. It can tell you whether you have a monosomy or one copy or three copies, a trisomy, but it's not, the microarray is not the test to actually do for the balanced or unbalanced translocations like I was showing earlier on the Down syndrome patient. it has the advantage of finding very tiny deletions or duplications. Now, this shows 300 kilobases, but honestly, there are uh, the probes are 1,000 long, and so if they if there's a copy number variant within a gene that's highly critical, the lab will report much smaller deletions, like down to 20 or 25,000 base pairs. An oligonucleotide microarray cannot give very low levels of mosaicism. So one of the things that we like to know with, let's say, Down syndrome is, number one, is it the standard trisomy 21? Is it the unbalanced translocation, which has impact as far as recurrence with the parent? Or is it mosaic? Like, is there a population of normal cells, 46XX versus 47XX with the extra 21? And of course, it can't tell anything about point mutations. So I'm going to go to two cases here to kind of discuss them and the key molecular tests that were done. A 2-month-old who was an ex-preemie, mom, had preeclampsia, C-section birth because of breech. Mom had a number of complications during the pregnancy, but overall was healthy. The baby had fetal lung maturity with the standard beta methasone. And the infant was AGA pretty much, and looked normal, normal physical exam. And but like 33-week preemies was a pokey eater, and so needed to have gavage feeds. So an NG tube was placed in the hospital, and the X-ray done to see where's the tip, and found a butterfly vertebrae. It was kind of odd. Nothing else was seen, but the staff decided to do a chromosome microarray as an initial test because we usually get a CMA first. And what it found is the child had a 754 kilobase deletion of the top part of chromosome 16. And these reports, you'll see that they have numbers, like one number to another number. And I'm going to show you something about how that works. And another key thing is that it talks about the genome build. And although we're now greater than 37 or HD19, seems like the chips are still back in the 2009 version. So the report will tell how many OMEM genes are within that sequence. I mean, that segment, pardon me. So the way that I analyze this, and there's other what we call genome browsers. This one is from University of California, Santa Cruz. And I mean, look how many organisms the are available, down to fish, I think, way down there. So we have to make sure that the correct build is here, and that's on the CMA report. And then I put chromosome 16, and then here's the numbers that are the so-called breakpoints. And then I generated this picture showing the top of 16, the P arm, and the bottom part of 16, the Q arm. And in this little red rectangle, it's expanded in this region. And all of these letter number deals, like the PPRT2, these are the genes that are within that region. And I programmed it so that it would search and say, okay, with the PRRT2, you have this particular disease, and you can click on it, and it will either link to OMEM, which is Online Mendelian Inheritance in Man, set up by Dr. McCusick way back in the 70s, now online through NIH and Johns Hopkins. And then also down here, there are gene review links. So you just click on this one, let's say, and you've got gene reviews. You click on this one and you go directly to OMEM. So this child actually has a known deletion syndrome called 16p11.2. And typically the children have intellectual disability, global delays. They can have congenital abnormalities, but generally butterfly vertebrae are not one of them. And interestingly, the children or adults who have a deletion in this region are obese, whereas the individuals who have a duplication of this same region are very, very thin. They have a way low BMI. So it's a kind of an interesting thing of a differential phenotype based on quantity of the genes. But basically, this is something that I would find if I did a chromosome microarray of a child coming into the office with intellectual disability or autism and was not expecting uh, the child to have this because, I mean, the child's 2 months of age. I mean, we're not going to see intellectual disability in a child of that age. And so this is totally unexpected. It's kind of TMI, too much information for the family at that juncture, because now the family has to deal with, well, will my child have all of these developmental and cognitive issues moving forward? And it doesn't answer why the child actually has the skeletal abnormality. So this was taken off the internet, a link to OMEM. There's a number for every condition. And basically, this is a curating all of the phenotype, all of the features that are seen in individuals who have this. But as you know, someone doesn't have to have every single one to clinch the diagnosis, may only have one or two. Uh, this is a really good resource if you come across an abnormal microarray. It's a British site, rarechromo.org. We call it unique. And they have uh, ever increasing number of articles that are readable for physicians, but they're also at a level that parents can understand for the most part, with pictures of patients, uh, diagrams of DNA, explanation of that, and then it kind of goes through step by step all of the issues that have been seen in individuals with these problems. And then this was a link to gene reviews, where the key gene that's felt to be responsible for intellectual disability, autism in these kids, is this PRRT2 gene. So what would be my plan at this point? Well, I would like to know whether it was inherited from the parents or not. And so possibilities is to draw a blood sample from parent and do fish testing and ask the question, is there one little light going on or are there two? It could be also a microarray. And part of the problem with this, in some respects, is that the parents are normal functioning or high functioning adults. They don't have any problems. So insurance is not going to want to cover this. And so it is an expense for the family. And the question is, was it inherited from a parent? Could be inherited from a parent who doesn't have any symptoms, which makes the whole situation pretty hard to interpret, uh, because some parents are asymptomatic, whereas some are not. Or sometimes these variants are de novo, meaning that they just happen by chance when the egg or sperm were created. Uh, And so what I decided to do is let's wait a while, see how the baby is progressing, and then since the infant was healthy when I saw him, uh, decide if any further testing is appropriate. This is a very interesting case. The child actually was hospitalized the end of last week for a second time. So some of you may have seen, probably a number of you have seen this child. So back in July last year, uh, the 2-year-old boy came through the emergency room, was found to have developmental delay globally, weakness. He wasn't walking at 24 months. And a number of regular labs and biochemical labs were done, including a CK that was sky high at 6,000. And I think it was a little higher during the hospitalization. Excuse me. So I saw him 2 months later in the Valleywise clinic, and he still was not walking. He had low tone, and remarkably, his CK was 51,000, had a high aldolase in congenital disorders of glycosylation or carbohydrate deficient transferrin. Typo. I'm a little dyslexic. That was normal. So my plan at that point, given that family history was negative, no one on that the mom uh, talked about on either side had any issues with, let's say, muscular dystrophy, delays, intellectual disability. I decided to do a chromosome microarray and panels for neuromuscular and metabolic disorders. And this is what came back, which was absolutely astonishing. There were no copy number variants found, so no deletions, no duplications. But the child had almost 1 billion base pairs that were homozygous, which is 25%, give or take, of the genome. So what this represents is first-degree consanguinity, incest, where the mother's either the mother's father and daughter are the parents, or brother and sister. This was not part of the history. Uh, and so when you have this extreme degree, it really, really increases the chance of autosomal recessive disorders. And this is the highlight down here, summarizing the gene mutations that were found on those two panels of this electron transport chain gene, uh, otherwise associated with what's called multiple acyl-CoA dehydrogenase deficiency, or glutaric aciduria type 2. It's recessive. The report shows the variant. So at position in the DNA 636, it should be an A, but it's a T. And then the protein is changed. It's homozygous. And it's classified as an uncertain variant as opposed to benign, which basically they're not going to report, or pathogenic, disease causing. There was one copy of this mutation in the AN05. Never heard of that gene. It was called pathogenic. But here's the key one, this SGCA gene. This was pathogenic and it was homozygous. Actually, I got these results back first, and I got the microarray back second. And then the child was also homozygous with the US of MIC U1 gene. So when I got back this microarray showing this high degree of consanguinity, it was kind of a no-brainer. We've got to do whole exome sequencing, got to see, does the child have more than one autosomal recessive disorder? There's a high likelihood of that. And basically, it came back showing the same results. Uh, when when I talked to the mom, we had already involved social services, and I had contacted Jody, and we talked to the legal department just to see what our responsibilities are as far as the safety of the mother. You know, she's clearly being abused. Um, you know, what is the deal there? And so the mother just basically said, you know, in a nutshell, when I was back in Mexico, this was a this is something that just happened in my family. You know, yes, my father, but this was years ago, or brothers and sisters uh, in my small agricultural community in Mexico somewhere. Well, it's not normal, obviously. Uh, well, you haven't seen your father for 5 years, so it's obviously not your father. Uh, who do you live with? Well, the baby's father is some guy who lives down the street, you know, completely unrelated to me. Well, that doesn't work. That's not how there's no explanation other than in this scenario that her brother and her are the parents. Um, and so we try to find out, okay, identify the father. Just tell us who the father is. And basically she didn't come up with a name of father. So in the whole exome sequencing, we were able to get a sample from the baby again and the mother. And so this shows that the pathogenic mutation in this SGCA came from mother and unknown because somebody else. And then the same thing with the MICU1 gene, mother and unknown, still a VUS. The ETFD1 gene that was homozygous was mother and unknown. And then the ANO5 was not from the mother. So that's without a father sample, we don't know, did it come from the baby's birth father, or did it happen by chance when the egg or sperm were created? So in other words, a de novo mutation. So what this just opened up something because I actually hadn't encountered this gene before. So like so many things, there's 20,000 genes and I know, you know, quite a few hundred of them, but the thing in genetics is we have to do a lot of reading and research to understand what we're dealing with. And so it codes for a protein called alpha, that's why it has an A, sarcoglycan. And interestingly, there's four genes, alpha, beta, delta, and gamma, which are represented in this picture here. So these four proteins associate in a tetramer with another protein, dystroglycan. This interacts with dystrophin, which is the muscular dystrophy, Duchenne's protein. They're in the membrane, and they all work to kind of stabilize the membrane and the extracellular matrix. This is just a three-dimensional representation of what the protein looks like. And so, interestingly, and this is something the neurologists know a lot more than I do, that this particular gene is the most common cause of limb-girdle muscular dystrophy, uh, abbreviated with these letters and numbers. And it's associated with proximal muscle weakness in the lower extremities. So presents with children who have delayed motor milestones, wing scapula, the calf muscles get way big, like Duchenne's. And occasionally, the children have cardiomyopathy. They have high CK, as this child does, and apparently is progressive. So delayed walking, but then later on, they lose the ability to walk. This is an OMIM printout. And so I did some reading here, and I came across a paper from 16 years ago or so, where in this picture you can see this adult's forearm, and right in this area here, they stuck a needle in, and they injected a adeno-associated virus that had attached to it the SGCA gene. And they injected a bunch of this hybrid virus, and they asked the question, did this virus, well, particularly did the gene get into the muscle cells, and did this have any effect? And what they showed is that they had a control. So on one side, they just gave saline, and on the other side, they gave the virus. And this is a staining uh, with antibody against the alpha sarcoglycan. And you can see that on the membranes of the muscle, it's highlighting, it's fluorescing. And here's a Western blot showing more thick signal in the treated. So this is kind of a demonstration of, like, why do we do gene testing? Well, number one, we want to know what does the child, what does the individual have? How can we then relate that to the child's present condition? Can we project over time how things are going to pan out? Uh, Compare and contrast, basically, our patient with other patients who are reported in the literature, or ones that we or our colleagues have seen together. And then what kind of therapy is available? Well, all kids are going to get, physical therapy if they're having delays, um, so they can achieve their maximum potential. But is there a specific therapy that's available? Uh, this is obviously very research based. And so essentially, um, try to get the mother back to discuss this. Well, are you interested in me contacting these doctors? If they're still, after 16 years, are they still at that institution? Are they still doing research into this? Is there any clinical study, like clinicaltrials.gov, through the NIH clearinghouse of things? Like, would you like me to investigate that to see if someone is interested in this, and could your child be included in a study? Is there something available now that could help your child? Uh, Unfortunately, the mother no-showed the appointment with me in January, no-showed with Dr. Escambe. And I think part of that is because of her social situation, and, denial of the incestual relationship that she was in, uh, her safety, how she's able to care for herself and her family. And so this basically is a very, very involved case, and shows all of the ancillary, all of the other things that are so important with our patients, that they're not just numbers and gene results, but they're families, they're individuals who have very extensive needs in many respects. And so my last slide is talking about whole exome sequencing versus whole genome sequencing. Uh, a whole genome sequencing is the ability to look at all of the base pairs of the genome, as opposed to the 2% in whole exome. And actually, it has a slightly increased pickup rate, even of the exons, than whole exome sequencing. And part of that is because it's able to find small deletions or insertions that are not possible to be seen in whole exome. I have a few cases like that. So it can look at introns, enhancers, these other regions that I was talking about. It can look at trinucleotide repeats, Fragile X it can detect, and it also includes the mitochondrial genome. Whereas with whole exome, if you want to look at the mitochondria, you have to ask for that separately. Part of the problem about doing whole genome testing is that we know, a fair amount about the exons. We know a fair amount about proteins, but there's still more to be learned than what we know now. But we have just massive ignorance about all these regulatory regions of the genome. So there isn't, a big enough supercomputer to really totally analyze. There's not enough knowledge about these intergenic, the regions between genes, for us to really understand what they mean. So in most cases, whole genome testing is not prime time, although we can order it from time to time, and it is the test that the state will pay for sick newborns. I have some slides that I put on about inborn errors of metabolism, but that's going to be a different talk. And how about I stop now, and if there's some questions, I'm more than happy to answer them.
Thank you, Dr. James. That was a good review of background in genetics and then where we are now. I don't see any questions in the chat at the moment. Um, if anyone has any questions, I welcome people unmuting themselves. And if not, I think that maybe. All right. Well, thank you so much. This has been a great presentation and really appreciate it. Thank you.
Absolutely. And have enjoyed working with everybody and look forward to continue interaction.
Same with you. Have a great morning.
You all too. Okay, bye-bye.